In a modern world of growing biotechnological innovations, manipulation of an organism's genetic codes has become more and more of a controversial topic. CRISPR, for example, is currently being widely disputed as being unethical. But what if there were other methods of genetic manipulation with less intrusive or controversial applications? The biological technique of optogenetics may be the most intriguing scientific breakthrough of this decade. Essentially, this technique manipulates neurons, a cell found in the brain, into synthesizing a protein known as an opsin. Unlike other proteins, opsins are unique in their light sensitivity as a result of their normal location being in the chromophore retinal found in photoreceptor cells of the retina. Put simply, optogenetics is essentially mind control on a neuronal level. By manipulating an organism's functions through genetic modification and light sensory stimulus of its neurons, optogenetics could essentially improve the lives of many patients suffering from Alzheimer's, stroke recovery, and Parkinson's disease. This manipulation takes place in neurons, cells found in the brain which are responsible for receiving sensory input from the external world, sending motor commands to our muscles, and transforming and relaying the electrical signals of these functions through the brain and nervous system. Neurons are made of three main structures: the dendrite, where a neuron receives input from other cells; axon, the output structure of the neuron, which sends an electrical message called an action potential throughout the entire axon when a neuron wants to communicate with another neuron; and the soma, where the nucleus lies, where the neuron's DNA is housed, and where proteins are made to be transported throughout the axon and dendrites. How then can neurons be genetically modified? Through the process of viral transfection, neurons can be genetically modified in order to make them susceptible to light manipulation. In transfection, the gene in an algae which codes for the specific light-responsive protein known as an opsin protein is isolated. First, nucleic acids are extracted from the opsin. Next, the nucleic acids are attached to a promoter, which causes recombinant DNA to be active in only one type of cell. It is then attached into a virus, which acts as a vector, though it is modified so that it can't cause disease. Next, it enters its host cell, a neuron, and injects it with its genetic information, forcing the neuron to incorporate its genetic code, causing it to begin making the protein that codes for the ion channel. When the neuron is activated by light, its ion channel will open, causing the neuron to fire an electrochemical impulse, which is then transferred to neurons of the same type. A promoter DNA region that only allows expression in specific cells is also included in the transfected genetic code to make sure the opsin protein is only produced in the desired cells. Opsins are membrane proteins with a molecular mass of 30 to 50 kilodaltons and are related to the protein moiety of the photoreceptive molecule rhodopsin. They are unique in that they typically act as light sensors in animals' brains and are specifically light-sensitive proteins. Due to their susceptibility to light waves, when opsins are inserted inside the nucleus and DNA of a neuron inside an organism's brain, specific hues of light, depending on what type of opsin is inserted, can be used to control a neuron and therefore its neurotransmitters in order to achieve different manipulative results. These, in turn, deliver the desired manipulated message to the organism's body, therefore controlling its functions. Different types of opsins also react to different types of light. Channel rhodopsin 2, for example, is a type of opsin in the subfamily of retinylidene proteins that function as light-gated ion channels in the neuron's cell membrane. They serve as sensory photoreceptors in unicellular green algae and contain a non-selective cation channel, again in the cell membrane, which opens in response to red light, allowing a flow of positive ions into the cell. This leads to the switching on of the neuron's functions. Halorhodopsin is another type of opsin with a light-gated ion pump. However, it is specific to chloride ions found in archaea, known as halobacteria, and opens in response to blue or yellow light. By opening its ion pump, which triggers an influx of negatively charged chlorine ions. This hyperpolarizes the neuron and causes it to act as an off switch to the neuron's functions. But what does the future hold for optogenetics? While optogenetic technology is still being further developed, researchers have already identified several potential applications for this technology in the medical field. For instance, optogenetics could be effectively used to fight Parkinson's disease. With wirelessly controlled microscopic implants, which could be inserted to illuminate selected brain neurons already genetically modified to be light-sensitive with LED light, they could potentially carry an internal drug delivery system to treat Parkinson's disease at its core. In the end, as scientific investigations begin to move more towards understanding the brain and the extent of its functions, using safe technology and non-intrusive methods of experimentation is becoming crucial in gaining further scientific knowledge. While this technique is still partially in its stages of experimentation, it's safe to say that with its absence of possibly unethical attributes and numerous applications, optogenetics may become the future for new knowledge of the human brain.